Hello, Living Environment Regents students. My name is Miss Ruth Tavares. I am a living environment teacher, have been doing this for 12 years with very high success pass rates, ranging from 99% to 82% pass rate, depending on the school I'm working in. But today is a very special video for my students as well. So this video is both for my current students and for students all around New York State. If you hear a difference in this video, it's because I have a super duper new microphone that's picking up everything, including the background, and I'll try to understand how to take care of that another time. And a very fancy PowerPoint upgrade, surprisingly, on my computer. But let's get started. So before we start, please remember to subscribe if you are, especially if you are a student who is trying to graduate, get those credits, and pass this Living Environment Regents for January, this January, coming up, January 2020, Living Environment. Please subscribe. I will give you information on my online course. My online course is for those students who have high need of the entire curriculum, and you'll be able to learn the entire curriculum in just about a week's worth of videos. Maybe you can binge watch it the same way you binge watch Netflix. You can binge watch my videos once you get my online course, which I will give you more information once it is all done. But you have to subscribe, and I promise you, you will pass with flying colors once you take my online course. So let's talk about what will be accomplished in this video. So there are two things for two different audiences. For my current students, you will get to do your vacation homework with me. When I was in high school, I wished that I had someone's help when I had all these packets. Hate that word. My students know I hate that word. But we were given packets after packets after packets for vacation homework. So much homework for vacation, but with me as your teacher, you get to do the vacation homework with me, along with the audience of YouTube and other students. So, for my online students, for those who are not my students, and you're all over New York State, this is a quick preview of what is to come for my online tutorials for the January 2020 exam. You're going to get good quality stuff with just the direct tutorial of what you need to know to pass. No fluff in between. Again, I've been doing this for 12 years. Now, some things to keep in mind. My current students, you need to do this to stay on point with this class. Don't let quarter two slip. This counts as an exam, an easy 100. As long as you have your packet and as long as you watch your video, this video, you should get 100. For my online students, my subscribers, this is a chance to review what you should have learned in the first quarter of school. So this is about 2 to 3 months worth of schoolwork. Some important dates to keep in mind for January 2020 Regents exams. Now, you might be taking more than just the Living Environment exam, but just to let you know, the Living Environment exam is January 21st. There is a good chunk of time until that time, a little less than a month, for you to take the Living Environment Regents, which is at 1:15 p.m., my favorite time. You get to sleep in a little, you get to come in later in the day, not necessarily close to the start of the exam, but you get a little bit of time for yourself in the morning to relax, to meditate, to get ready, look over your notes. Afternoon exams are the best. And here is a quick snapshot of the upcoming Regents exams for January 2020. Good luck, everyone, and I hope that you pass. I hope that you find some good tutorials out there for you to help you with these Regents exams. I hope your school that you're in gave you the right resources to help you pass these incredibly difficult, high, heavy content exams in such a short period of time, unless you were taking on a workshop or you got a tutor. So good luck to everyone, especially my period 9 group of students who have been so amazing in going to class. So what we will cover in this video, so of course, like in all of my videos, we'll be going over Regents style questions on various topics, covering quarter 1 and quarter 2 of living environment curriculum, as I said before. So this involves the cell theory, the cell, the animal and plant cell, the compound microscope, and the one of the 4 mandated Regents labs, diffusion through a membrane. This one focuses on the cell membrane, and you'll see examples, and hopefully you remember and hopefully you did this, because not every teacher covers 100% of the curriculum. You're supposed to do this lab. You're not even supposed to take the Regents unless you do this lab. And if you see my previous videos, I have videos specifically just on these labs. So let's get ready. Are you ready? So let's go. So here's number 1 of your vacation homework packet. Again, I hate that word, but question number 1. All of the following are true regarding cells except. So we have to keep in mind that this is saying except, meaning all of these are true, but they want you to choose the answer that is not true about cells. So let's see number 1. All cells have genetic material? Yes, they do. All cells have genetic material, which is DNA. DNA is a molecule that is in your cells in the nucleus, that is the blueprint of life. It's the reason you are who you are. B, all cells have cell walls. This is not correct. All cells have cell walls? No. Only plant cells and some bacteria or prokaryotic cells have cell walls. Animal cells do not have cell walls. Our cells don't have cell walls. So right away we know that the answer to number 1 is B. Number 2, what is a similarity between all bacteria and plant cells? Well, let's take a look at the answers. So this is saying what is a similarity. If this were one of my 1-hour videos, I would go back to our chart that explains the differences between eukaryotic and prokaryotic cells. One of them being that eukaryotic cells have organelles and prokaryotic cells do not have membrane-bound organelles. So A is wrong, where that they both have a nucleus. No, a bacteria does not have a nucleus. A bacteria is a eukaryotic cell. So they do not have a nucleus. They do have genetic material. So similarity, they are both composed of cells. Yes, that's true. A bacteria is a cell and plants are made up of cells. So that's already the answer. 2 is B. They are both composed of cells. C, let's just take a look just for the heck of it, just for laughs and kicks. They both have chloroplasts? No. Chloroplast is where photosynthesis happens. Photosynthesis is when you take energy from the sun and convert it into sugar for the plant. So chloroplast is, happens really only in plant cells. It's only found in plant cells. Some bacteria do photosynthesize, but for you to know, particularly chloroplasts are found in plant cells. D, they both lack a cell wall? No, plant cells, as we said before, have cell walls. So 2 is B. Number 3, which sequence of terms is in the correct order from simplest to most complex? So this is talking about the smallest to the largest. And in the cell theory, we know that the basic unit of living thing is the cell. So the smallest thing from what I see here of our choices is the cell. So we already know that B and D is not the answer. So let's take a look at A. So we have A, cells. When you clump cells together, you get tissues. From tissues, you get organs. And from organs, you get organ system. This is the correct form. Cells to tissue, organ systems? No, organs come first, then organ systems. So the right answer for number 3 is A. Number 4, Which organism is considered an exception to the cell theory because it has a non-cellular structure? No matter what or how they ask the question, the exception to the cell theory is the virus. Scientists have been arguing this for years. Now, I haven't looked at recent research about viruses and if they're considered part of the cell theory, but for many, many, many years, when viruses were discovered, they became the exception of the cell theory because they don't really look like a cell, but they have DNA material and they multiply once they invade a cell. So the exception of the cell theory will always be a virus. Next, number 5. Living things contain units of structure and function that arise from pre-existing cells. So this is basically saying, part of the cell theory, that cells come from pre-existing cells. This statement best describes the cell theory. So 5 is A. Sometimes the best thing for you to do is to cover the answers and see if you can come up with the answer without the options, the multiple choice answers. Number 6, during an experiment you want to view the nucleus of a cell. How would you most easily view this organelle? So if you've used a microscope before and you've looked at, let's say, your cheek cells, you took a toothpick and you took samples of the inside of your cheek cells and you mounted it on a wet mouse slide and you viewed it, you can see under the microscope, under maybe objective number 10, that you were able to see your nucleus. So the microscope is something that you would need to view the nucleus. You cannot see it with the naked eye. You cannot see it with a magnifying glass. And you could not see the nucleus with any of these. No, you can see the you can see the nucleus with the microscope. Definitely. So 6 is A. Number 7. Which structure is best observed using a compound light microscope? So yes, you can see a cell. No, you can't see a virus. That's too tiny. No, you can't see a DNA sequence. That's too tiny. And no, you cannot see the inner surface of a mitochondria. That is too tiny for a microscope to capture, a compound light microscope. So the answer to number 7 is also A. So just to let you know, it does not matter the pattern that you're beginning to see. If you have 10 questions that all of them are A, and you know for a fact that all the answers are A, do not change because you feel like, wait a minute, they can all be A. Don't worry about the pattern. Just focus on what is the confident, correct, first instinctual answer. Okay, so this diagram pertains to both 8 and 9, and in the case you forget to read the directions, you want to make sure that if you don't know what you're looking at, read and see if there's any clue given to you in the directions. So here we have, base your answers to 2 questions, so questions 8 and 9, on the diagram below, which represents a typical green plant cell, and on your knowledge of biology. So this is a plant cell, and we know this is a plant cell because it has 2 layers. It has a rectangular shape or squared shape. It has a large central vacuole, and it has chloroplasts, these large globule things and inside their chloroplasts. Now, one of the things that I hate about the living environment exam Regents and all Regents is that there is no color. Now, if this were a plant cell, they'd color this green and have it color coded. So, number 8, which organelle is a cell wall? The cell wall is D. So 8 is D. The outermost layer of a plant cell is the cell wall. 9, which one is the mitochondria? Whenever you see a bean-like structure with these squiggly lines inside, this is a very good magnified picture, but that is the mitochondria. So we have 1, 2, 3, 4 mitochondria. So the mitochondria is C, and that is the powerhouse of the cell where cells make energy. Number 10, here we go, another diagram, another picture. Always read the directions and see if there's any clue. Base your answer to the following question on the diagram below represents a plant cell. So here's a plant cell, and even though it's a little blurry, you can see that it has two layers. It's semi-rectangular in shape, even though the edges are a little bit smoother. It has large central vacuoles, and then these are chloroplasts. So let's take a look at the question. Which cell structure functions as a storage site for waste, water, and food? This will always be the definition of the function of the vacuole. The vacuole in plants is much larger than in animal cells because plants need to conserve water, food, and waste. They're organisms that don't move. They are still in one spot. So they have to conserve as much energy and water as possible. So the answer to 10 is D. D is pointing to one of the two vacuoles, and that is the organelle that holds waste, water, and food. Number 11. The diagram below represents a cell in a human body. And these are my favorite cells. They're animal cells. They're round in shape. And here we have a ribosome. One is pointing to the ribosome, which makes proteins. 2 is pointing to the nucleus, which is where you find DNA, controls the cell. 3 is pointing to the mitochondria, as we said before, and it makes energy for the cell in the form of ATP. And 4 is the outermost layer of the animal cell, the human cell. And that is the cell membrane, controls what enters and leaves the cell, which we'll get to it more. We'll get to it more of that in the future of this video. So let's see the question now that we've described what we're looking at. Which statement concerning the structures within this cell is accurate? Number 1, structure A is a chloroplast? No. First of all, this isn't a plant cell. Chloroplasts are only found in plant cells. So 1 is definitely, 11 is not A. B, structure 2, which is the nucleus, is a vacuole? No. Don't even play me. No, I don't think so. C, structure 3, which is the mitochondria. Okay, is the mitochondria where respiration takes place. Cellular respiration. Cellular respiration is the name that's given to the biochemical process that makes energy, that makes mitochondria the powerhouse. So 11 indeed is C. This is correct. And let's just for fun and kicks, of course, structure 4 is the cell membrane. That's correct, which provides rigid support for the cell. Incorrect. Rigid support is the cell wall. Cell wall gives rigid support for the cell. So this is incorrect. Next. 12. Mitochondria provide ribosomes with energy. The mitochondria gives energy. So in the form of ATP. So let's see if we have an answer that correctly fits what we just said. A, ATP for protein synthesis? Yes. Yes, 12 is another A answer. ATP is the energy made by mitochondria to make proteins, and the process of making proteins is called protein synthesis. Number 13. The diagram below represents a cell, an animal cell. Which letter indicates a specific structure where most hereditary mutations occur? So hereditary mutations is when the DNA is incorrectly made. When instead of a nitrogenous A base, there's a T. There's a substitution or an inversion, some kind of mutation. There will be a future video discussing just mutations. So hereditary mutations, hereditary DNA, where is the DNA found in a cell? In the nucleus, which is C. C is pointing to the middle round structure, and that is where you find DNA, in choice C. Number 14. The diagram below represents a cell that produces digestive enzymes. So enzymes are also proteins. Which cellular structure would be most likely the location for synthesis of these enzymes? I added this one. Now this is a Regents question and I typed this in for my students. We just started the topic recently, so in the future when they see this question again, you guys will not have this as a hint. But where are proteins made, or what organelle helps the process of protein synthesis, of making proteins? The ribosome. The ribosome sometimes is free floating in the cytoplasm. the cytoplasm being the jelly-like structure in between all these spaces here. So 1 is the ribosome, and that is where protein synthesis happens in the ribosome, and 14 is A. Look at all these A's, but that's okay, because that's the right answer. Okay, so here we have a microscope. Let's take a look at the question. A student studied the upper layer of cells of a tissue sample under the high power objective of the compound light microscope shown below. Which part of the microscope is the objective lens? So this one isn't necessarily a Regents question, but you, as a student, must know what the objective lens is. Where is it? So the objective lens are here. They're usually 3 or 4. So D is pointing to one of the 2 objective lens. So 15 is D. Excuse me. 16. A student is viewing a plant stem cross-section using a compound light microscope. What parts of the microscope should the student use to bring the image into focus? So this particular student is already has the objective lens set. They already have the stage set, and they're asking, they just want to have the image focus. So you don't want to move the stage. All you want to do is move what's called the fine adjustment. This one helps you with clarity. This one helps you to focus and see in clarity what the field view has. So that is E. So the only one that has E is B. So yeah, you want to move, I guess for this one, they do want you to move the stage just slightly, but the one that you would move particularly for focus is the fine adjustment, which is E. This is the fine adjustment. Fine adjustment is the smaller of the 2 knobs. This is the coarse adjustment, which moves the stage up and down, and the small one is the fine adjustment, which just helps it to focus. Okay, this one is just about memorization. Here we have what's called a wet mount slide. You have the glass slide. You have your specimen that you're going to look at with the drop of water or the drop of stain, whatever it may be. And here you have a cover slip. And here you have a arrow stating that you're about to let this go and let it drop. So let's read the question. The diagram below shows how a cover slip should be lowered onto some single-celled organisms during the preparation of a wet mount. Why is this a preferred procedure? This, my friends, is to prevent air bubbles from happening. Now, you'll always have air bubbles, but this is to prevent the air bubbles from happening. So 17 is D. The possibility of trapping air bubbles is reduced, but you will still have air bubbles. Okay, so this is the part of microscope questions where a lot of students get confused. When you move the slide on the stage, you should know that everything is opposite. If you move your slide to the right, this image is going to move to the left. If you move the slide to the left, this image is going to move to the right. If you move the slide towards you, the image will move away from you, and so on and so on. So everything is the opposite effect. So here, let's read the question. In which direction should the slide be moved to center the cell in the microscope field? So we want to center this cell in the middle. So let's see. If we move the slide to the right and up, so if we go right and up, this one is going to go left and down. So it's going to end up here. So that's not it. Next. Oh, right. So it's going to move there and up. So, oh, even I got confused. See? So for number 18, A is the correct answer. So if you move the slide to the right, it's going to move left. If you're going to move the slide up, it's going to move to towards you. So yeah, this one will end up in the middle, 18A. This one at least gives you numbers to refer to. Same question. In which direction should the slide be moved to center the cell in the field view? So we want it to be centered. So we want it to move this way. So you have to move the slide the opposite way. So you're going to move it towards 4. So 19 is D, toward 4. Same question. Now I put several of these because these can get confusing if you forget, which I did myself, and I'm the teacher here. Oh my goodness. In which direction should the slide be moved on the microscope stage to center the cell in the field view? So you want to center this cell. So you want this cell to move this way. That means that you have to move your slide towards you. So you wanted to move it towards C. So 20 is C, towards C. All right, now here is one of the points that I want to make about taking a long test. We've already gone through over 5-10 minutes of this video, and I'm getting tired. So this is a good time to take a break if you need to. Pause the video, take a break, walk around, drink some water, wash your face, but come back fresh and rejuvenated, because you want to make sure that you build stamina and that you understand how to answer these questions for when the Regents comes, okay? And if you're in school and you're actually taking the exam, you need to ask to go to the bathroom and wash your face, drink some water. If your school allows you to bring some kind of drink, you can take it outside and take a quick sip of, if it's a juice or a soda or whatever the case may be, they'll give you a nice boost of energy. So take a break and then come back with fresh eyes. All right, guys, welcome back. If you took a break, I know I did. I drank a nice tall glass of cold water. You gotta hydrate yourself. If you don't drink water, go ahead online and Google how to find out what's the proper amount of water for you to drink every day. Some of you might drink too much water, and some of you might not drink enough water, and that is necessary for your brain to function at its capacity. You want to be on point when you take this exam. Okay, so 21. Now, just to let you know, this particular packet that I made for my students has 35 questions. So we're almost to the end, and after a certain number, I think after 23, we're going to work on some short answer questions, meaning that they're not multiple choice, and they're particularly pertaining to the diffusion through a membrane lab. So here we go. Let's continue. 21. A student is observing a temporary wet mount of an onion epidermis under the low power objective of a compound light microscope. So they're right now on a low power objective, which would be the most appropriate procedure for the student to follow in order to view the wet mount under the high power objective. So before I read A, B, and C, and D, you don't want to move the slide. I'm sorry, you don't want to move the stage. Once you set the focus, you should not move the stage anymore. All you have to do is move the objective and adjust the fine adjustment, because once you go higher, now let me go back to a couple of questions where there's a compound microscope. If you go to a higher objective lens and you move the stage up, you can crush the slide and your specimen, because this is long, and if you move this up, this can actually touch and crush the slide. So definitely don't choose something that asks for you to move the objective lens. Let's see. Remove the cover slip? Absolutely not. Add another layer of onion epidermis? No, this is ridiculous. Add a drop of water to the specimen? No. Increase the amount of light passing through the specimen? Yes, because when you go to a higher objective lens, you lose the amount of light that passes through the lenses, because you're going through thicker lenses to get higher resolution here, a more magnified view. So you're losing light. So you want to increase the amount of light with the diaphragm. Next, 22. Which substance is used as a stain to make it easier to observe a wet mount of a cheek cell with a compound light microscope. Now that is methylene blue. So 22 is B, methylene blue. That's what we used as a stain. 23. Which laboratory procedure is represented in the diagram below? So here we are placing either a stain or salt, or this could be stain, salt, or sugar. It's looking like a stain right now, and we are letting that flow through the cover slip and using a paper towel to suck up the water inside of the cover slip. You don't want to open that. You can ruin your specimen. You can lose your specimen. It could be tiny cells, and if you lift the cover slip, it's just, you're going to lose your specimen. So this creates a flow, and it's going to let this run through, and the paper towel is going to pick up whatever's being released, and then the cells get stained. So let's see, placing a cover slip over a specimen? No. Removing a cover slip from a slide? No. Adding stain to a slide without removing the cover slip? Yes. 23 is C. Again, for fun and kicks, let's see, reducing the size of air bubbles? No. This is when we drop, this is when it's talking about the 45-degree drop on the slide. Okay, 24. Now, when you see questions like this, you tend to get nervous. You right away assume, I can't answer this. What is all this? I got to read so much. What's this 1 hour later? Why are there 4 pictures here? If you did this lab, you know exactly what's happening. This is a diffusion through a membrane lab. Now, we're in the short responses here. My iPad is charging, and that's where I would have come up and done a better job at what I'm about to do. So I have to leave my full screen, and I'm going to draw as best as I can, guys. As best as I can. Let's do red, yeah. What's going on here? So I'm going to go ahead and read the question, and I'm going to explain what this is, and then we're going to answer the question. Base your answer to the following question on the information and diagram below and on your knowledge of biology. 2 models of a cell were made with the dialysis tubing. Dialysis tubing is this paper bag. It looks like a paper bag that you wet and got soft, and you tied one end and you put glucose and starch in it, and you tie the other end, and then it looked like this kind of candy wrapper thing. And you're testing for diffusion, the movement of molecules from high concentration to low concentration. So this experiment was to try and see what went through the cell and out of this artificial cell. And this dialysis tubing represents, it's a simulation of a cell membrane. So 2 models of a cell were made with dialysis tubing and placed in 2 beakers of fluid, A and B, each containing starch indicator solution. Starch indicator solution, ladies and gentlemen, is iodine. That brown dark dye liquid that you used for this lab. As represented in the diagram below, enzyme Z was added to the artificial cell in beaker B. The solution outside each cell was tested for the presence of sugar. Initially, no sugar was present in the solution outside each cell. The results after 1 hour are represented below. So here we have the starch indicator, the beaker, starch solution, milky white. And then after an hour, this starch solution turned black, blue-black, right? We saw a little blue. Now here, beaker B, starch indicator, beaker, starch solution with enzyme Z, and then here we have that there's sugar present here, and here starch solution, again, it turned black and blue. So we have a similar result for beaker A. So this is one beaker, and this is a different beaker, and you have the same result. Now here it says, state one reason for the color change in beaker A after 1 hour. So the solution is, the reason is diffusion. Diffusion is the reason why there was a color change. Now, don't just write diffusion in your answer, okay? What I want you to write is a full a full correct sentence. Now, what you should write, and I'm going to minimize the font to 11, is, so state one reason for the color change. The color blue diffusion, because that is why there was a color change, because the iodine, the starch indicator, diffused into, it was small enough, the molecule was small enough to go from high concentration to low concentration into the bag, and it touched the starch, which is the milky white substance, and after an hour it turned the bag blue-black. Okay, let's go to the next question, and let me get my drawing. Let's just see what this looks like. Oh, no. Okay. So here, this is going to be answering 25 and 26, this diagram here. Now, if you didn't do this lab, my explanation could be enough. It could be enough. If not, you have to ask your science teacher, make sure you give them a donut and coffee and beg them to please do this lab, so that it's a stinky lab, but you have to do it to understand and see. Just a heads up, I will be doing it'll be part of my class, my online class. So base your answer to questions 25 and 26 on the diagram below and on your knowledge of biology. The diagram illustrates what happens when a particular solution is added to a wet mount slide containing red onion cells. Now, my students, I'm sorry I put you through that smell, but we had to do it. This is when we took the red, what I call purple onion, and we took a piece of the skin and we observed it under the microscope. Cells being observed under the compound light microscope. So, identify a process that caused the change in the cell. So what are we looking at here? We have the onion cell in its full glory, and we added something that shrank the cell membrane, because remember, the onion cell is a plant cell. So on the outermost layer you have the cell wall, and then the inner layer, the next layer, is a cell membrane, which it shrank. And everything in between, you're going to have the cytoplasm, the jelly-like substance that contains all the organelles. So the cell membrane and cytoplasm shrank. Identify a process that caused the change in the solution. Diffusion. Diffusion. Okay? So actually, let me type that in. So diffusion. That's what caused the change. What kind of thing diffused? What kind of molecule? Salt. Salt, when diffused in the cell from high to low concentration, it dehydrated the cell. The water molecules left in order to let the salt in, and hence it made the cell membrane shrink and the cytoplasm shrink. Okay, identify a process that caused the change. Oh, it's the same question. Oh, Miss T. Miss T. Oh, don't forget to subscribe. I did that by accident, but why not? Copy and paste. Guys, students, just write diffusion twice. Sorry. Okay, here we have a cell, and it's a cell that shrank, much smaller. So here it says identify one likely substance in the environment of the cell that causes response. Hello. Now, it's not diffusion because it's asking for the substance. I just needed the text box. So then it's in the flow of questions. You could say salt, but if you add salt or sugar to a cell, this is what's going to happen, because it's going to diffuse into the cell and the water molecules diffuse out. So the answer, just pick one. And for the time being, since we're doing this, put salt, just salt. But for you to know, you can also put sugar. We're almost at the end. As you can see, we're almost at the end. Okay, so let's take a look at this. We know that this is the 45-degree angle drop to prevent air bubbles. We know that. So Let's see what it says. State one reason why this is recommended procedure for placing a cover slip. Oh my gosh. Oh my gosh. Now I'm going to use that text box of diffusion again, but don't type diffusion. Don't write diffusion. This is to state one reason, reason being. Now remember, when you're taking an exam, you're going to write full sentences. So yes, Mr. Barris, this procedure is recommended is to avoid air bubbles. Please, please, please be articulate and precise. Next, oh, and for this one, I am going to use black, and let's make this a little, oh, yeah, a little bigger. So, let's undo that. So, here we have a big question, 29, one of those questions that you're like, what do I have to read? Why are there so many diagrams? Why is there a key? Calm down. Start from the top and work your way to the bottom. The diagram below represents a laboratory setup used to demonstrate the movement of molecules across a selective permeable membrane. That's talking about diffusion and cell membrane. And notice how it's the same lab, the starch and glucose. And this is supposedly the bag, selective permeable membrane, the bag that we use that represents the cell membrane. So the large molecule here, the large one, oh, looks like googly eyes. Oh. So the large molecules is starch, and we know that. The small one, small molecules is glucose. And then the selective permeable bag that has these microscopic holes where certain molecules can flow through. In the diagram below, draw the 5 starch and the 12 glucose molecules to show where they most likely be located after 15 minutes. So let's do the starch first. So actually no, because my pen is already very close to what a glucose molecule looks like. So after 15 minutes, these glucose molecules went through because they were small enough. They were small enough to go and pass through the selective permeable membrane and live in this new space from high concentration to low concentration. So we are going to do our 12 glucose molecules on this side. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Very good. Now I'm going to change my pen to thin, and we are going to add our starch molecules. Now these starch molecules were not able to pass through. In the case you're like, how am I supposed to know that? You are supposed to know that from your lab, and you can also see that this molecule is too big to fit through there. So we're going to draw our 5 starch molecules on this side, and you want to make sure that it looks like I tried my best. If I had my iPad charged and connected, it would have automatically made these beautiful, wonderful circles from my crappy, discombobulated circles. So this is what you should have, guys. So take your time with Regents questions, understand, remember what you did in class. Hopefully you did this lab in class. Next, another one. So let's see. So here we have glucose. This is the lab. We know that glucose diffused out because the molecule is small enough. The molecule was small enough to pass through the bag. So let's read the question. In the diagram below, draw, and remember that when it says draw, you do it in pencil, in the expected location of the glucose molecules after 20 minutes. So the glucose was in the bag. After 20 minutes, it should be out the bag. So we are going to draw our glucose molecules outside of the bag, none inside of the bag. And I'm not really going to really worry about how many glucose molecules there are, but this is what you should see. This is what you should have in your papers, my students. You should not have any of the glucose molecules drawn inside of that bag. Next. 31, we're almost at the end, guys. I'm sorry I exceeded my 25-minute mark that I promised you, but at least it's not an hour. And if it's an hour, I'm sorry, but it shouldn't be more than that. You're going to be done with science, and you're going to be all caught up, and you're going to have 100 on your test when you hand in your answer sheet. Because remember, oh no, I'm collecting both. Okay. 31. An experiment set up using a model cell is shown below. The dialysis tubing, blah, blah, blah. State what cell structure the dialysis tubing represents. The dialysis tubing, we know that it represents the cell membrane because the cell membrane, bring that up for you, for those OCD people. The dialysis tubing represents the cell membrane. The outermost layer of this plant cell, now remember this is the onion cell, so an onion cell is a plant cell. The outermost layer is the cell. Done with 32, so you want to make sure that you have that nice and clear. Next, 33. Okay, remember, read from the top to the bottom. Base your answer to the following question on the information and diagram below and on your knowledge of biology. A wet mount of red onion cells as seen with a compound light microscope is shown below. Okay, very good. In the space below, sketch what cell A would look like after after you add salt. So I am going to have fun and know that this is an onion cell that kind of looks like a plum color. So when you add salt to the onion cell, we know that it dehydrates the cell membrane and the cytoplasm. So this is the cell wall that stays as is, and then the cell membrane shrinks. It no longer has the full effect. It shrinks. So we are going to shade in, of course, the cell membrane and the cytoplasm. You're going to fill that in. You're going to shade it in as best as you can. You only need a pencil. I like using colors to just remind ourselves that the cell membrane is purple or red, the onion cell, I mean, and the cell membrane dehydrated itself to make room for the. So this is, you don't have to label, but this is the cell wall. This is the cell membrane, and this is the cytoplasm. Just make it thinner. This is the cytoplasm. But you don't need to have that. You don't need to have the labels. They just wanted you to draw what happens after you add the salt. It shrinks. The cell membrane and the cytoplasm shrink. Shrink, shrink, shrink. Next. Okay, start from the top, work your way to the bottom. 2 questions left, almost done. A solution containing both starch and glucose, okay, still the diffusion through a membrane lab, was placed inside the model cell represented below. The model cell was then placed in a beaker containing distilled water. So here we have the dialysis tubing. We tied one end, then we added starch and glucose, tie the other end, put it in a beaker in water. Identify one specific substance that should have been added to the distilled water so that observations regarding movement of starch could be made. So what they should have done and added to the water is the iodine. So the starch indicator. So a specific substance, one specific substance